in pursuit of health and wisdom. Sapio with Buck Joffrey.
Welcome back to Sapio with Buck Joffrey. I'm Buck Joffrey, and today I have got a phenomenal guest here, Dr. Hans Keirstead. He's a chairman of the board of Immunis, which is his company. He is an internationally recognized stem cell expert and really successful entrepreneur. He's known for developing treatments for quadriplegic spinal cord injury, cancer therapies for melanoma, brain and ovarian cancers, and multi-pathogen vaccines. He's also advanced therapeutic research for motor neuron diseases, retinal diseases, immune disorders, and age-related immune decline. He's a serial entrepreneur now, a guy who started out as a pure academic, and now he's working his way through the business world to offer this stuff to real people. So, Hans, thanks so much for joining me on Sapio.
Real pleasure.
So I'm going to start off right away and kind of just get into this. I mean, your story's super interesting, but there's so much material. I just want to make sure we get through. So, here's the thing. One of the reasons I really wanted you on, Hans, is we talked about this offline, is right now you want to call it longevity, you want to call it, whatever you want to call it, this space is a multi-billion-dollar industry that has a lot of really cool and interesting stuff that's really coming down the pipeline, but it is also mixed in with an enormous amount of snake oil and an enormous amount of charlatans who are capitalizing on that. And so it was really important for me to actually bring somebody on and really talk about what's real and what's not in this space. And so I appreciate your being here for that purpose. But let's start with some basics. I mean, for people who are out there and they're hearing about stem cells everywhere, frankly, let's start with the reality. What are stem cells, and why are they promising in treating disease and potentially helping with longevity as well?
Well, it's a really good question. And, following up on how you introduced this, I would like to say that when I started this field and started in this field of stem cell research some 25, 30 years ago, 9 out of 10 stories were snake oil. And today, 9 out of 10 stories are snake oil.
Right.
So it's very, very difficult for lay public, of course, to know what's real and what's not. And there's a lot of easy ways for you to figure that out. If you put in Hans Keirstead, FDA cease and desist, and surf for whether or not the FDA has served me with a cease and desist letter, you'll find that they have not. You'll find that most stem cell people purporting to be stem cell researchers and purveying stem cell goods have actually been told to shut their shop down by the FDA. So there's a couple things you can do to discern it. Certainly go talk to, experts about it. You've got to do that work because it's potentially extraordinarily dangerous for you to be putting a stem cell treatment in your body that's not regulated. So one has to ask themselves, why is it that a company like in the United States or Europe or an advanced nation would ever skirt the FDA? Why would they not embrace the FDA? Because any drug that goes to the FDA reaches the more people that in a faster speed than a drug that does not. And that's just simply true. The companies grow bigger, the money's bigger, your reach is greater, you're treating more people. Why would you avoid it? And the only reason you'd avoid it is because you can't meet those qualifications. And if you can't, there's no human that should be taking it. So just a quick word on that. But what can stem cells do? The field of stem cell research really started off with the idea that a stem cell could actually become any cell in your body, which we certainly know is true, and therefore can replace tissue that's lost or nurse it to stop it from dying. And although that's true, stem cells do have the ability to differentiate into various cell types, enabling regeneration, repair of damaged tissues and organs. It's actually been learned over the last 20, 30 years of this that an exogenous cell, extra cells like stem cells transplanted into the body, they're kicked out. Even if it's not a rejection issue, just putting more cells into an organ, they get kicked out. Somehow the body knows how to regulate the cell content It knows what is not endogenous and growing from the current tissues. It knows what is different from the current tissues, and it does a very good job of booting that out. So the tissue regeneration is less from the cells themselves becoming new tissue, but rather the secretome that they release, the sum total of all of the proteins, growth factors, cytokines, chemokines, all these regenerative, soup that they make. It's these little proteins that are touching your endogenous cells, the cells that you have that are maybe dying or needing repair, and turning them younger. So we do know that, stem cell products do very fundamentally change the way that your genetics are expressed, gene expression, or for the deep, schooled audience amongst you, epigenetic changes. But it's mostly that nursing factor, the secretions that they make. They also have another thing altogether, which is immune modulation. Stem cell therapies can modulate the immune system, and it's almost impossible to put stem cells or their secretions into a body and not modulate the immune system. And that reduction of chronic inflammation and the associated damage is broad spectrum and touches all organs, not only the one that's perhaps hurting. And I guess the last thing about how stem cells are being used these days would be as a targeting mechanism. Young cells like stem cells, they have just an innate desire to go to a damaged area. So wherever you put them in the body, they find all the damaged areas and they go there. And what they'll normally do without any other intervention is just secrete locally and nurse the dying or hurt tissue. But if you engineer them to perhaps deliver a drug, that's another kind of trick, new way of getting a drug or a therapy delivered to the site of the damage. So those are mostly the ways that stem cells are being used these days.
I think the critical piece to what you're saying, if I may summarize and simplify, contrary to popular belief, it is not the cells themselves. It's not like you've got these cells. The idea is people think that you've got stem cells, sort of these cells that can turn into anything. They're like these cells that are like little baby cells that can turn into anything, and that you're just injecting them into an injured organ or a severed spinal cord, and boom, you're getting these stem cells to turn into the kind of cells that you want. And that's not actually what's happening. It's actually several other mechanisms that are involved with, some of the magical, not magical, but seemingly magical mechanisms of stem cells to release various growth factors to go where they need to go in order to perform their function. So I think that's actually a really, really critical difference in what people think. And frankly, that might be one of the things that people use as sort of a radar to try to understand whether something is real or not. Because, you still see, you go to a med spa and they're giving you stem cells, right? So, okay. So with that being said, just a few practical applications, if you could talk about in that context so we can illustrate it. I mentioned to you that a couple decades ago, I was a neurosurgical resident, and before that, a medical student with a lot of interest in neuroscience and neuroregeneration. And at that time, there was lots of these experiments. Nothing worked. Bottom line is nothing worked in terms of neuroregeneration. And I think it has to do with the fact that we actually thought those cells were going to do something. We thought those little cells were going to turn into something when in fact, we just talked about how they don't. You actually have done some pretty significant research in animals in quadriplegics and have gotten neuroregeneration. So what is the mechanism where you take a severed spinal cord and you're actually able to regenerate neurons then?
In order to get a cell that you transplant into a human to incorporate into the system, you've got to do a couple things. First of all, you have to have it in purity. You need a pure population. If you're going to fix a spinal cord, you're going to want to put in spinal cord cells. If putting a stem cell in there is the worst thing you could possibly do. You don't want to put heart cells into the spinal cord. You don't want to go put toenails in the spinal cord. So the first thing is purity. And my group is the leading group in the world for high purity derivation of cell populations from stem cells. So we were the first, and actually to this date, the only group in the world and I've got around 150 peer-reviewed publications to this effect, where we can take stem cells and make them into a spinal cord cell type or other cell types, but for this discussion, spinal cord, and in purity, 99.6% pure. And if you do that, that's one thing. You've basically addressed the deficit by putting in cells that are deficit. So you got to make those cells, and can you do it in purity? So that's one thing.
So just to clarify, you need to give them a head start. You can't start these cells from nothing and tell them go become a neuron or an axon. You have to turn them into neurons and axons, young neurons and axons, and then deliver them first.
That's correct. Because if you think about it, if you put a stem cell that can become anything in the body into an area of injury, that stem cell is going to smell, taste, eat its environment, right? Mm-hmm. And that environment is saying, make scar. Because that's what happens in a human. When you hurt your brain or spinal cord, you make scar. It does not regenerate, you make scar. So if you put more stem cells in there, they will just become more scar. Mm-hmm. So you have to differentiate them in purity to the thing that you need. And then the second thing that you have to do is make sure that they're young, because it's only young cells that can incorporate. Old cells cannot. They just get kicked out right away by your body. So we're the only group in the world, actually, that's been able to make young cells of a lineage-specific progeny. So that means we're not putting toenails in the spinal cord, we're putting spinal cord cells. In this case, oligodendrocytes. I had discovered that after spinal cord injury, that it's not only the spinal cord axons or wires going up and down that are broken. That happens, maybe 5% of the spinal cord gets broken like that. But the penumbra, the surround, all of the axons, the wires going up and down, connecting what's below to what's above to your brain, they're functionally intact. But I should say they're anatomically intact. They're still there, but functionally they might as well be cut because they're stripped of an insulator. Like a wire in a wall has an insulator around it. If you take that insulator away, it short circuits. That's exactly what the spinal cord does. You have this maybe 5% in the middle that is blown away by the traumatic injury, but then over the course of a few weeks, it expands to be 100% of the spinal cord becomes naked. All of those wires are stripped of their insulator. I discovered that and then generated from stem cells the insulating cell type. It's called an oligodendrocyte. So we, for the first time in the world, made high purity human oligodendrocytes, these insulating cells of the human central nervous system, from stem cells, and we kept them in a developmental stage where they were young. Now, that was the first time in the world anybody had made a high purity, 99.6% of anything from any stem cell population. So that was the start of my career in high purity, because high purity means useful. So in that case, that very rare instance, we were able to transplant cells made from stem cells into the spinal cords of rodents and then humans. They actually restored all of those insulation around all of those wires, myelin on axons, and basically restored the function of the majority of the spinal cord again. And 26 humans that had full quadriplegia, meaning no sensory and no motor below the chin, with 50% of our dose, 100% of these people had full use of their arms and feet, interestingly, with motor and sensory. And then we gave 100% of the dose, and they got that again, but started to get anecdotal evidence of bladder, bowel, and sexual function back. So really powerful. But that was one of the rare instances where cellular replacement worked. Right. And it was only because we were the only group in the world, and to this day, we're the leader in the world of making high purity populations. And that's a real challenge for the stem cell field.
I'm curious, why is that? I mean, it seems like a pretty remarkable find. Why has that not become sort of standard? Why are we not seeing quadriplegics, with this kind of recovery?
Yeah, Buck, it's one of the great learnings of my career, and one of the great sadnesses. There's not enough spinal cord injured patients. If you
So there's
depending on
not
how
the
you classify,
funding to make this a standard treatment. Is that?
there would be a lot of funding if there was a market. Basically, there's only about 18,000 spinal cord injuries a year in the United States. And if you had a drug that fixed spinal cord injury and it worked on 100% of them, that would be about a $300 million annual market. And big pharma doesn't touch anything that's below $1 billion annual market. So, once you get past the heart money, into the smart money in building a biotechnology where you're needing big checks like 10 million, 20 million, $50 million checks in order to run these $100 million clinical trials and things like that. It's smart money. And those individuals that decide if they're going to cut you that check, they're financial analysts and scientific analysts, and financially, it actually doesn't make sense. Wow. Because they'll never get their money back. The market is so small that it would take years and years and years and years for them to even break even. So because it's an orphan indication, it's very much laid to the side. So great therapy, very little market.
Yeah.
So that was a very hard thing.
So that's not available anywhere to somebody who's.
It was after those great results were found, and honestly, nothing negative, just all phenomenally good. The problem is that a chief finance officer walked into the board one day of the company running it and said, "Hey, board members, why are we even doing this? Financially, it makes no sense." Yeah. And so they dropped the program.
That sucks.
Yeah. Well, so that's to your point though, very rare instance of where the cells do the work. 99% of the time, it's not the cells. The cells are living for a little while, die off, and they secrete the nursing factors, the secretome.
That's right. And I want to get into secretomes a lot more here in a moment with longevity in particular. But before we go there, I want to even talk to you about research on cancer, because I think this there's probably a good leeway into the longevity application. You've had success. Tell us a little bit about the research in using stem cells for cancer. And now this is a situation where you're really focusing on, those secretomes, right?
Yeah.
And first of all, why don't you define secretome before we go any further? Because we've used it a few times.
Sure. The secretome is the sum total of the things that are secreted, released by a cell. When you're a little human growing up in your mother, one organ at a time generally is being made. They don't generally overlap. They do, of course, a little bit, but at any one time, there's one organ, being made, and another and another. Of course, they overlap in different areas, but generally speaking, there's a lot of concentration on one organ at a time during development. And what's happening there is all of the youngsters, like the young brain cells when it's developing, or the young heart cells when it's developing, they secrete hundreds of factors and bathe their neighbors, and they all say, "Let's become heart." And that's what drives a concerted development towards making the heart. So it's the secretions that are growth factors, they're genetic influencers that cause a unified formation and regenerative or generative event within development.
Let me ask you this, because, just going back to things that are commercially available, left and right, I am seeing med spas offering quote unquote exosomes. Can you talk about what those are? Is there any legitimacy to this? And go from there.
Stem cells, 9 out of 10 stories are snake oil. It's more of a marketing campaign than a real thing.
Yeah.
In those 9 out of 10, of course, it's a real thing in the 1 out of 10. Same thing can be said of exosomes. Exosomes are part of the secretome. They are released, but they're not a little protein, a growth factor that just has to be released. As soon as it touches a cell, it affects an action. An exosome is a bundle of little genetic modifiers that are all in a little ravioli. They're in a little membrane.
Bunch of DNA.
It's actually things that influence the DNA.
Okay.
Yeah. And that's not DNA itself, but it's things that cause genetic alteration of what's turned on and what's turned off. And they're contained in a little ravioli, a little lipid ball. And they have to diffuse across fuse to a membrane, dump the contents into the cell. Then you have to have a diffusion within the cell into the nucleus or mitochondrial DNA, the areas, the organs of the cell that contain the DNA, and then affect a change. So there's a lot of steps there. So therefore, there's a huge loss. If you had started with 100 little exosome things, you might have one hit the target. So exosomes are complicated. They're actually a fairly narrow regenerative set of functions, where the rest of the secretome is very broad and a lot easier. It's a single step of a little protein binding to a receptor. Boom, you get an effect. There's a whole bunch of steps with exosomes. So exosomes are a small component. They generally comprise about 0.5% of a secretome. They were kind of neat and sexy when they first were isolated, because it's a way of genetically engineering somebody without putting in little toxic things to their genes. It's a kind of a natural way of genetic engineering. However, the efficacy is extremely poor, and they can really control what they do. So they get them from young cells like stem cells, hoping that they'll be good.
Yeah.
But I have to say that it's about the same thing. 9 out of 10 things you hear about exosomes is more of a marketing campaign than a reality.
Yeah, and I would imagine even, just looking at that, like you go to any place that has exosomes, I mean, where are you getting them from? Maybe 1 out of 100 places might actually give you a little bit of exosome. The rest of it might just be a bunch of saline with dead proteins, or not dead proteins, but denatured proteins that are not beneficial. So, okay, well that's good to know.
And Buck, can I add one thing there?
Yeah.
If you're ever looking at an exosome content, I'm talking to the audience here.
Yeah.
An exosome product, you have to ask yourself, you have to understand that exosome is genetically modifying you. That's pretty hardcore.
Yeah.
Very broadly, it's going to go to every cell in your body and genetically modify you. And why is that purveyor of the exosome treatment? Why are they skipping FDA regulated clinical trials on something that is so extraordinarily powerful in engineering you? You want to be very cautious of this stuff.
Yeah. Yeah, absolutely. Okay, so let's take that message back to secretomes, and then I presume this is where cancer applications come in, right?
Well, this is where actually our longevity and health span drug comes in.
I knew that part, but so how does cancer work? How do you use this for cancer? How do you use stem cells for cancer?
Yeah, so my stem cells for cancer, I'm running in a company called Avita Biomedical, where I'm the chairman and CEO and founder of the company. We are in phase 3 clinical trials. Phase 1 tests safety, and we've done that. Phase 2 tests whether it works, and we've done that. Phase 3 tests whether, there's a side effect if you do it on 7 populations and males and females and pregnancy, etc. You're looking for contraindications so that you can say on the bottle, do not take if you're taking that drug. So it's low, much lower risk. We've already shown that it works. So Avita Biomedical, we have a stem cell approach to cancer and we've published this. We've run clinical trials in the United States in 9 hospitals in a phase 2, and we've shown a 50% enhanced survival above and beyond the greatest current standard of care for cancer in the world.
Which cancer?
This is in glioblastoma and in melanoma.
Okay.
And we've actually also shown it in several other cancers. So it's all cancers, but we're only moving forward right now with glioblastoma and melanoma because they're very, very expensive trials in a phase 3 setting. But what we do in this case is that we take advantage of a discovery that I didn't make, but I was the first in the world, myself and my team, to take advantage of it and make it clinically and commercially viable. So somebody at Harvard and Stanford and a couple other places in the world, all around the same time, they found what was irrefutably the most important discovery in cancer. And that was the discovery of the tumor initiating cell. It's called a TIC, tumor initiating cell. Some people call it a cancer stem cell. And these TICs have 3 functions. They actually grow the whole tumor. That is an extraordinary discovery. It's 1% of every single tumor type, all tumors. 1% is the TIC, the tumor initiating cell, and it grows the whole tumor. The second thing they do is they can leave the tumor, migrate, that's called metastasis, and make distant tumors. And then the third thing, perhaps most insidiously, one of them can leave the tumor and just fall asleep, cover themselves in sugar like a little blanket, making themselves invisible to the immune system. And they sit there for 2 to 7 years. They don't divide, they don't do anything. They're completely invisible. And they sit in your large blood vessel or in your bone marrow, and they just sit there quietly like little sleeper cells. And then for reasons unknown, they wake up 2 to 7 years later. Now, think about the patient. Patient thinks they're in remission. They've maybe done chemotherapy and irradiation or checkpoint inhibitors.
Right.
And now they have an exacerbation, a recurrence of their cancer. That is because those little sleeper ticks woke up again. And your ticks are different from mine. Every human has thousands of unique mutations. So when these cells were first discovered, I'm a stem cell scientist, and my group is the best in the world at high purity culturing of human stem cells. It's irrefutable. We have like 150 peer-reviewed publications showing that we are. It's not just me. So, when I saw that, I grabbed that technology, and now what we do is when somebody has, say, glioblastoma, brain cancer, they're getting a resection. Most cancer patients, almost all of them, have a chunk of the cancer taken out. We have a little tiny vial in the surgery room, and they just plop a little cubic millimeter or cubic centimeter into that little carrier liquid that we provide. It gets sent to us, and we get a little blood from the patient too. From the tumor, we extract that 1% and we expand it in purity. So now we have your 150 million of your pure ticks, not 99% of the tumor that looks a lot like you. It's your own hair, gut, skin, cartilage, slightly mutated, but it looks like you. The tick does not. So we are the only group in the world to be able to isolate the ticks. It's a stem cell. And then what we do is we grind that up and feed it to the general of your immune system. It's called a dendritic cell. And we get that from a little blood draw. We isolate your dendritic cells, again, amplify them up. And now we have your dendritic cells. That's like the general of your army of your immune system. And we feed them the ground up ticks. And they are all those little proteins and bits and pieces of the tumor initiating cell are eaten by the dendritic cell and expressed on the surface. So the drug is actually just your own immune system. And we put it back into you, that your immune system that has been educated to go kill ticks. And when your dendritic cells are reintroduced back into you, what it does is that general of the army tells the immune system, go kill this thing, the ticks. So that kills the growing tumors, it kills metastasis, and it kills the sleepers. So we get about a 50% enhancement of survival. And I'm just delighted. I can't tell you how just wonderful it is for me and my staff to be surviving 50% more humans with this drug that costs us extremely little to make and has no side effects.
I'm curious, so that doesn't cost a lot to do that? Is it on an individual basis?
No, it does not. It costs so little that not only is it affordable to Americans, Europeans, first world nations, but it is also affordable to lower and middle income countries. For the company, that opens up a huge market because now we can look at 75% of the world, which is lower and middle income country, and provide them with an affordable cancer treatment.
Okay. So the tick itself it's a type of cancer stem cell, right?
Correct. It is the cancer stem cell,
And so what you guys were able to do is essentially multiply those up so that you get a whole bunch of those and get like a personalized immune reaction and get a bunch of antibodies. Personalized to them.
Antibodies and cellular toxicity. That dendritic cell that has been educated to recognize the ticks, the cancer stem cells, we put that back in the body. Not the stem cells, but only your own tumor initiating cell that's been educated in a dish. And it says, go kill this thing. And it tells your immune system. And you don't do this naturally. You have to do it in a dish. And then reintroduce your own immune system back into yourself. Just these generals of the immune system. And then it's like the general telling its army, its air force, its SEALs division, go kill the ticks. And then a huge immune response. It's not only antibodies, it's also Th1, Th17 cellular toxicity, and they go kill them. And it's the highest survival that's ever been reported from a therapy, and it has no side effects.
Right. Let's go on now to, I guess, the drum roll, I guess, is the for longevity, and which is
very fun.
mostly kind of what I had kind of followed up what you were
Yep.
doing there, and this is where those secretomes come into play. Where
Correct.
now again, just a reminder to people that this is not the cells themselves; these are proteins, growth factors, all those nice things that these stem cells seem to make. So, how do the secretomes affect potentially issues of longevity?
So I've got to give you a little bit of schooling in order to answer that question.
Sure.
But it's very short.
Perfect.
So, okay, your health span and your lifespan
Yes.
is more about, it's not really about your genetics, but it's about how your genes are actually expressed.
Right. Your
Think about
epigenetics.
this.
Right.
You have the same genes in you when you were a baby as you do now, but clearly you look a lot different. So the genetics doesn't really drive much. It's about, the estimations from scientists are 10 to 30% of your lifespan and health span is due to your genes, but about 70 to 90% of it is due to what genes are turned on and what are turned off.
Mm-hmm.
And when you look at what are the things that change genetic expression in humans, the number one thing is the immune system. The immune system has a greater effect on what genes are turned on and turned off than any other thing in your body, any other thing that a human experiences. The immune system is the dominant way of changing genetic expression. So how do you drug the immune system, right? How do you tweak that thing? Every drug you've ever taken is a single active ingredient, one single molecule that does one very specific thing and nothing else. Now, that doesn't work in the immune system, hence the paucity of drugs in the immune system. And the reason is that your immune system has tons of redundancy. For everything the immune system does, there's 5 or 50 things pushing it and 5 or 50 things pulling it. So those 10 to 100 things can't be drugged with a single drug. So, when I was thinking about how we modulate the immune system, because clearly if we want to have an impact on health span, lifespan, I theorize that we are going to have to modulate the immune system, but how do you do it? I was involved in a lot of government think tanks about secretomes.
Mm-hmm.
And it started with botanicals, interestingly, because, plants have very powerful drugs in them, and there are multiple drugs, but that was never accepted by the FDA's because of batch-to-batch variability. You boil a pound of willow bark, and you get a different amount of drug every time you do it. It's kind of like terroir in wine. Same field, same winemaker. Every year the wine tastes different because of the sun and the rain and the soil. So, secretomes are very hard to make from cells, except my team happens to be the best in the world at growing high purity cells. And if we can make very, very high purity human immune system precursors, then we could make a secretome that is an immune modulating secretome. Because the secretome of a stem cell is really good for making stem cells, but it has nothing to do with your immune system. A secretome from a cardiomyocyte, young heart cells, they're really, really good at making and repairing heart, but they have nothing to do with your immune system. They don't have anything to do with stem cells. So you can see that the sum total of what is secreted by a cell, what is that stuff? What is that secretome? It's actually extraordinarily tightly tied to whatever cell produced it. So what we did is we made a very defined human cell population that releases immune system secretome. So it is the only population in the world, and we are the best at making high purity populations, so we can make it the same every time. That means there's no batch to batch variability that is significant. And the FDA made us do multiple batches and say, completely different starting populations on different times. You got to start from scratch, but I want to see that every batch you make is the same or with an acceptable degree of minor variation. So we were able to do that, and we're the only group in the world that's been able to do that. The only other secretome story in the world that's really big and making waves amongst us, cutting edge researchers is mesenchymal stromal stem cells, or MSCs. You get them from the bone marrow, but you got to drill into somebody's bone marrow. That hurts. It's expensive. And then pull out some bone marrow, isolate the stem cells, mesenchymal stromal stem cells, and then make them into something. But now you have to put it back into the same person.
Yeah.
So it's not commercially viable because you got to say, oh, Buck, you want to feel
Yeah.
a little bit better? Maybe you don't have anything wrong with you right now, but you want to just like, be
Yeah.
younger.
Yeah.
Okay, I just need to do a $350,000 procedure on you that you're actually probably not going to notice a whole lot of because you don't have any problems right now, but it'll make you younger. It'll stave off things. You're not going to do it. And it hurts. It's very difficult, can't be scaled. With Immunis, a company that I'm the chairman of the board, it's just, I tell you, I'm so excited about this story. What we've done is made a defined human population of that secretes every single factor that's relevant to immune system development and health because of the high purity cell population that we've created here. We make them by the billions. We farm the factors they secrete. The product is only the little proteins of this that are secreted. And yes, they contain a little bit of exosomes in there as well, but we've defined every single factor in the secretome. There's nothing bad. There's everything that you had when you were younger, making young immune system cells. When you're 27, you birth your immune system every 2 weeks. When you're 57, you're birthing it every several years. Like you're making fewer and fewer immune cells as you age. And what happens is that means you have fewer youngsters in your immune system, in your blood, and therefore you have fewer secretomes, right? Those little proteins, these 440 factors. A 50-year-old has a fraction of what a 27-year-old has. So my hypothesis here was that, let's restore that content, those nursing factors. And we know what those nursing factors do. They make the immune system young again. They make it divide better, roll into tissues better, function better, signal better, healthier and younger. So we made this stuff and we handed it out to 7 of the world's leaders in aging. So, when you age, what happens to you? It's almost all immune mediated. So your muscles shrink. 100% of humans have muscle shrinkage. That's actually immune mediated. It's your immune system screwing up and getting old and angry in 100% of humans that causes muscle growth to decrease dramatically.
So sarcopenia is it's a
Yes.
Immune regulated. How does that happen? I mean, just broadly,
Sure.
from an
Yeah.
immune standpoint.
Yeah, the secretome from your immune system includes a whole bunch of muscle modulating and in fact muscle satellite cell. They're like the stem cell of the muscle. They're really not a stem cell. Stem means they can make everything in your body. These are little progenitors that can only make muscle. They're called satellite cells. But for all intents and purposes, a lot of people will call them muscle stem cells. So they're the things that have to be stimulated and to divide and fuse together to make more muscle. It's your immune system that controls satellite cell division. So if you don't have those nursing factors, the amount of satellite cells that actually respond to exercise are a fraction of what they were when you're younger. So when you age, you get smaller muscles. That's immune mediated. When you age, you get arterial stiffness. That's stroke and heart
that's
attack.
definitely immune mediated, right?
Yeah, that's immune mediated. When you age, you get neuroinflammation. That is a necessary condition before you get Alzheimer's and Parkinson and multiple sclerosis. When you age, you lose your adaptive immune response, clearly immune mediated. That's your ability to see and fight a new bug. When you age, you heal much poorer. Your healing, your burn responses and healing, that's immune mediated. And when you age, you also lose your metabolism. That's also immune mediated. So what we did is we took this immune secretome, young, healthy immune, 440 factors, and we gave it to 7 different laboratories, and each one of those manifestations of aging. And in every single laboratory, we saw tremendous betterment of old mice. We went forward with muscle atrophy, and because you look at all these different things like arterial stiffness and wound healing and neuroinflammation, adaptive immune response, metabolism, and muscle, and the 2 that really pop out for market reasons and the best selling drugs in the world right now, they're metabolic and they're metabolic drugs. like GLP-1s.
Sure.
They're selling 2 to 5 billion a month. They're the
Yeah.
largest selling drug ever in human history. And it's true that they burn fat, but they also burn
A lot
muscle.
of muscle, yeah.
Yeah, when they cause weight loss, 30% of the weight loss is muscle. And now, when you're older, you do not want to lose muscle. I'm a neuroscientist, and I can tell you absolutely matter of fact that muscle makes something called brain-derived neurotrophic factor that goes into your brain and causes cognition. It causes plasticity and memory. And old people have less of that, right? They
Yeah.
have trouble remembering, naturally aging. If it's really bad, they have Alzheimer's. It's muscle that has a lot to do with that. So you do not want to lose your muscle. Also, of course, your cardiovascular and your health and your ability to move and toilet and clean yourself, shower, feed yourself, all of that requires your musculature to work. So we ended up doing a first human clinical trial in muscle in humans. And I tell you, Buck, like, I've done this a few times, and it is the most exciting and also scary time of my career when we first administer a brand new drug into humans. Because and here, think about it, we're actually poking the immune system 440 times. So what could go wrong? A whole lot.
Yeah, right.
So we did tons of work prior to make sure that nothing should go wrong. We had hundreds of doctors opining with us, and the FDA, of course. These are all the possible things. And we ran those down and said, well, it doesn't do that. Here's another horrible outcome. No, it doesn't do that. Until we exhausted all those horrible outcomes. And then we administered it to humans. And I'm very, very pleased to say that all the data is coming back now. And from this phase 1/2A, which means safety plus does it work? And here's what we found. First and foremost, no side effects. Like, no side effects. It's extraordinary. Nothing attributable to our drug. That's just amazing. That's mind-blowing. GLP-1s, for example, cause really, really bad gut problems and things like that. But we have no side effects. 100% of the humans are growing muscle. They're getting stronger muscle, and they can walk further and faster within 6 minutes. That's called the 6-minute walk test. And it is an accredited quality of life measurement. That's probably the most important outcome, because it means that these humans aren't just looking better. It's one thing to look like Arnold Schwarzenegger when he was younger, but it's another thing to be able to lift like him.
Sure.
So does it have any functional outcome? And yes, it does. These people are walking faster. And in fact, when we compare it with a group of similarly aged individuals that are subject to three times a week of exercise for 6 months, our drug causes functional motor improvement that it's better than 6 months of exercise. In addition to that, it's causing their blood level, inflammatory level, to drop about in half. So, you can age somebody. You can take some blood and pretty accurately age them on how inflamed their immune system is.
Yeah.
The older you get, the more inflamed it is. And we dropped the inflammation in about half. So, putting a very broad, category onto this, these 70-year-olds have an immune system of about a 45-year-old now. That's probably responsible, the reduction in basal inflammation, for a 50 to 70% reduction in pain. They also have a near elimination of cartilage degeneration. Old people chew up their own cartilage, and especially our population who's immobilized because knee osteoarthritis. We use that to, get them. They're older and they're immobilized because of their knees hurting. And we're not treating the knee. We're just using it like an immobilization. It's easier than finding 70-year-olds wearing casts.
How is the treatment administered, Hans? Is it an injection? Is it IV infusions or what?
It's a subcutaneous. I'm sorry, it's a intramuscular injection, and we're backing off now to subcutaneous. We just maximize the effect by going straight into a muscle, and now that we see that it works so well, we're going to back off and just do sub-Q, and then try nasal inhalants and stuff like that.
Yeah, and in the studies that you have, were these administered, weekly, daily? How are they administered?
They're administered every week for 4 weeks. And what we saw was even 3 months later, the benefits were still increasing.
Right.
So the level of reduction of the immune response was still getting lower. The level of walking speed was still getting better. The muscles were getting stronger. They were getting bigger still. So I imagine that this is going to be, I'm just guessing here. We don't have the data at all, but just, I'm feeling it out for what I see from the humans and knowing a lot about human physiology in this sector. I would guess at this time that people will receive a shot every 6 months of this stuff just to keep their immune system strong and keep your muscle up. And the other thing that we saw that I really think is important is, in our animal studies, we had tremendous fat reduction. And the humans are also reporting a lot of benefit to their metabolism. Like, for instance, when you take the blood from an older animal or human, a mouse or human, you can see the biochemical markers of a poor metabolism. So basically, as your metabolism drops with age, you don't chew things up as much when you're eating and digesting them. And you have these big, what are called toxic lipids or bad lipids. And they're an indicator of a bad immune system. I'm sorry, it's a bad immune system, but also a bad metabolism. So the people are getting fatter because their metabolism's lower and their bloodstream, you can detect that with these markers of bad lipids. And in these humans, we're seeing a near elimination of their bad lipids. Mm-hmm. So that's showing that we're, between the animal work and the human work, we are shrinking fat, but we're building muscle and no side effects. Yeah. So a really, really hot drug. We're right in the hottest area of the market right now. Super excited to be able to help people.
You're actually in so many different areas there. I mean, obviously weight loss, sarcopenia. You're also in pain. Like, I was sitting here with my I've had back surgery 3 times and I'm 50, and I'm just sitting here in pain thinking, hey, Hans, you got any of those vials ready for me?
Yeah.
And let me ask you this, from an FDA indication standpoint, what will the indications, do you have an idea what they'll be?
Yeah. Yep. Like, what our phase 1/2A was muscle only. So it's muscle atrophy. So this will be a drug that abates and actually reverses muscle atrophy. But because we saw such tremendous signal in metabolism as well, fat reduction, our phase 2 clinical trial is doing both metabolism and muscle. So if that thing works as well as our phase 1/2A worked, and as well as if we just repeat what we've already seen in a phase 2 setting, double-blinded, randomized, placebo-controlled setting of a phase 2, then our label will be fat reduction, metabolic betterment with muscle function enhancement.
And no reason to, any reason not to throw in pain in there? Because that would be in
a clinical trial, you have to pick your outcome measures, and the FDA likes you to pick very minimally and very narrow. And these clinical trials are very expensive. So this phase 2 will be my 18th clinical trial that me and my team have, I've led. We're very good at this, and we managed to get the FDA to broaden it out to a lot of outcome measures. We're going to include pain in there. So we probably will be able to say decreases pain. We're grading as many as we can, but for sure, we're going to be running down metabolic betterment, that's fat reduction, with muscle growth. And we should be able to say pain, cartilage degeneration, et cetera, as well.
Do you have any sense of timelines for this? I know that's very hard to
It's hard. I always get in trouble when I forecast these things, but I can tell you that we're just finishing up our phase 1/2A right now. We did 3 cohorts of increasing doses. And the good news is that our phase 1/2A is now de-risked, because we have a cohort. We actually have 2 of them that have no side effects and tremendous efficacy. So we'll move into a phase 2. We'll apply in about 2 months. And now we'll probably get approval within a couple months. However, the average is 14 months. So it could take us 14 months of going back and forth with the FDA. That's the average time it takes. We might do it in 2 or 4 months, but if we did it in 2 to 4 months, as I suspect we will, that means in the latter quarter of this year, we'll be starting our phase 2 clinical trial in muscle and metabolism. That'll take about 2 years, probably shorter. Again, I think will be done in a year, but probably, well, I'm being conservative and saying 2 years, two to 2.5 years. And then there will be a phase 3 that'll take probably 3 years. Again, that's very conservative, could be 1 or 2. But in the meantime, there are several countries that have approached me for expedited commercial approval of this drug because it is so broad functioning and it has no side effects. So I will be working with several regulatory agencies around the world to seek fast track. Like, for example, I actually helped put in place some, one of several people, but, I think enabling, to put in place in Japan a regenerative medicine track that allows you to skip phase 3 clinical trials if you have great phase 2 clinical trials in a stem cell therapy. Well, this is a stem cell therapy. So we might be able to skip the phase 3 and then go for full commercial approval and then sell it from that country by mail order, for example. And so, some countries it'll be, several years. Other countries it'll only be, 2 to 3 years. So, it's an exciting path ahead of us.
How expensive do you anticipate this drug being?
I have a policy here. I have a very, very firm drive, and sometimes it drives my manufacturing team mad. But basically, with all of the drugs that I have developed over the course of my career, and certainly this one, we put a lot of effort into making this thing extraordinarily inexpensive.
Yeah.
I anticipate that this drug is going to be able to be sold in lower and middle income countries for just a couple dollars while making huge gross margins. Of course, it'll be sold for much more in advanced nations, but the cost of this drug is so inexpensive that it will be very, very low priced where it needs to be, because it's a real mandate of mine that I don't think that advanced nations have any more right to medicines than lower and middle income nations and poor people. So I try to develop the drugs that I'm leading in order to be accessible to everybody, and that basically means inexpensive. So I'm working very, very closely with several lower and middle income countries in order to expedite commercial approval of this drug and provide it to them at an extremely low cost so that they can treat the people in need there.
Fantastic. Well, it's interesting because I was just thinking about, I did an entire sort of solo podcast a couple months ago on this concept of inflammaging, right?
Yes.
And
exactly.
ultimately this is a kind of drug for inflammaging, right? That's kind of what
exactly.
This is.
I can't state that enough. And thank you for saying that. This is a new drug in a new drug category, and the only drug in the world that targets the immune system in a broad spectrum manner.
Yeah.
So I think we have something here that is very, very special, and that is played out. Like I'm just the company is doing extremely well right now. Interest from monetary supporters, from big pharma partners. I've never seen such interest in any company. And it's because the sector is so massive, so highly valued, and frankly, the benefit to humans is extraordinary. In advanced nations, of course, we can make the modifications to our behaviors and what we eat, et cetera, to help our lifespans. And when a drug like this becomes available, we'll all be taking it. But you think about, the continent of Africa, where the average age is 36. Right. Think about India, where the average age is just in the 40s. What the governments there are trying to do is just simply make their population live longer so they can pay more taxes and make jobs.
Yeah.
Because that turns that desperate economy into a thriving one and increases the standard of living for everyone if there's health and jobs and taxes and money to build infrastructure. So right from the, macroeconomics, right down to just you in your own life. This is a drug that has extraordinarily far reach, and it's all natural, it's all human, it's all physiologically balanced, and it's inexpensive without side effects. I think this thing's got some real legs.
Well, I want to thank you so much for being on. The company again is Immunis. Hans Keirstead, thank you again for being on Sapio, and we'll be following you very closely.
Thank you so much, and stay healthy, everyone.
Thanks for listening to Sapio with Buck Joffrey. A quick reminder that while I am, in fact, a surgeon, nothing I say should be construed as medical advice. Now, make sure to include your physician in any medical decisions you make. And also, if you're enjoying the show, please make sure to show your support with a like, share, or subscribe. Until next time, this is Buck Joffrey for Sapio with Buck Joffrey.